Today we will cover some of the altered metabolism in the pancreatic cancer and at the same time this TME metabolism that affects or that we can say help the altered metabolism in the pancreatic cancers or pancreatic ductal adenocarcinoma. So we will start with the panel A that discussed about what are the reprogramming that occurs within the pancreatic cancers. So first the changes that occurs first and the important changes that occurs is the uh, utilization of the glucose. So glucose are being utilized in the cancer cells mainly in this HBP pathway that helps in the formation of O-glycan and also this nucleotide formations through this PPP pathway. And as a mean of byproduct, what it will release is it will release the lactate that will helps in the change in the TME, it is tumor microenvironment, in terms of reducing their acidicity or the pH of this tumor microenvironment. That will further helps the tumor cells in maintaining its reprogramming stage in a way that will helps them in the tumor progression. So this is about the glucose pathway, and in normal cells, this glucose pathway will form glycosylases, and thereby it goes into the mitochondria for the ATP productions. And whereas in this case, the anabolic metabolism takes over, or in the pancreatic cancers, the anabolic metabolism takes over. Other changes that occurs are in the mitochondria. That is, it will trigger this oxophonous pathway, and during this what it will does is it will utilize the components metabolites from the TCA cycle and also from this malate aspartate shuttle or MAS to form the building blocks such as metabolites, lipids, amino acids, and not only from these pathways from the mitochondria, these building blocks are formed. These building blocks uh, are also been contributed by autophagy, which what it will in autophagy the organelles within the cells are being recycled so that they can maintain their tumor progression, and simultaneously the macropinocytosis also helps in maintaining this building block, also supports this building block. And what are the components in the macropinocytes that we will discuss in the panel B. So all this these three main pathways helps in making the building these metabolites, lipids and amino acids that will helps the tumor cells to maintain their reprogrammed state. And how the tumor microenvironment helps in this reprogramming is mentioned in this panel B. So one of the important factors that helps one of the important factors in the tumor microenvironment that contributes towards this PDAC progression is this cancer associated fibroblast. So what it will does is it will release some of the exosomes, and for those metabolites will be utilized by the pancreatic cancer cells for maintaining their tumor progressions state. And since uh, since one of the major contributors in the metabolites are the amino acids, so there are some pools that are being specially maintained or mainly contributed by some of the tumor microenvironment factors. For example, collagens helps in maintaining the pool of proline, whereas hyaluronic acids helps in maintaining the pool of glycan, and this macropinocytosis helps in maintaining the pool of alanine and LPA. LP is not the basic amino acid, but it is some of the combined form of these metabolites. At the same time, this macropinosynthesis, what it will does is that it will release this deoxycytidine, and this is also released by TAM, that is tumor associated macrophages, and we have discussed about this tumor associated macrophages in our previous video. So this deoxycytidine, it will competes with this gemcitabine, which is a chemoresistant drug, so that it will reduce the effect of this gemcitabine and prevent the cancer cells from the impact of this gemcitabine. In other ways, this deoxycytidine reduce the effect of this gemcitabine. And how these tumor associated macrophages are formed or differentiated from the monocytes is through the release of this lactate and GM CSF. So they will help in differentiation of these monocytes into this M2 type macrophages, which has the pro-tumorigenic properties. And the neurons that are present within the pancreatic cancer near the pancreatic cancer cells helps in providing the serine pools. So some of the amino acids pools are already discussed here, such as proline, glycinac, alanine, and serine, and other things are being contributed by the other factors such as metabolites or exosomes from the cancer-associated fibroblast, and change reprogramming within the pancreatic cancer cells. And that will helps in maintaining the tumor cells. That helps in maintaining the tumor cells. It's reprogrammed state. So I hope you got some basic information about what are the changes occurs within the pancreatic tumor cells and how the tumor microenvironment helps in maintaining those kind of altered metabolism. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.